Hi everybody. So as you remember, this is going to be the second part of this discussion, and we are going to actually dig into the central dogma, which again explains how DNA is encoded into proteins. As a reminder, there are two processes to the central dogma. The first one is when DNA goes to RNA. Specifically, we're going to look at messenger RNA, and this is called transcription. And the second part, taking messenger RNA to protein, is called translation. Transcription happens in the nucleus. Translation happens in the cytoplasm. So protein production always starts with DNA, and what it essentially starts with is a gene. That is a small region of DNA that's in part of a chromosome, right? The sequence of DNA in each gene is what's going to encode or be the specific protein that we make. So in order for proteins to be produced, it requires this kind of intermediate molecule called RNA. RNA is a nucleic acid, as a review from what we looked at the beginning of the semester when we looked at biomolecules. But how are they different from DNA? Well, a big part, if you look, here are the areas on the bottom. And when we're talking about 5', 3', we're looking at this as a very first carbon because this is where the base would be attached if we had the sugar. So that's 1, that's 2, that's 3, 4, and 5. So on the second one, you'll notice in DNA, it's called deoxyribonucleic acid because D means it's without an oxygen, which is there. So that's one way that it's different, right? And when we talked about 3', I said when you looked at 3', there's always a hydrogen as the last atom. Well, it's true, but if you look at it, it's fully the OH that's there at the end. And then essentially what will happen up here is you'll get that phosphate that's there. So when we look at the sugar, that's the difference on the sugar. Now, a big thing that we'll notice, and you can easily tell for sequence of an RNA if you have DNA or RNA, because if you have DNA, you have thymine, and if you have RNA, you have uracil. So when we look at this, G's and C's still base pair with each other, but anywhere you have a T, you would now have a U in your messenger RNA. So if you have a DNA sequence, this is our DNA, that has C, G, T, and A, your messenger RNA would then be still G, it would be C. Where T is, it would be A, but where A is, it would now be U. So you can put this together the same way as you do your complementary DNA sequence. It's just wherever you would have a T, you'd replace it with a U. Now that makes a big difference in terms of what we have here. Because what we have, if you look here, messenger RNA tends to be single-stranded. And then DNA, we know it's double-stranded. And it also is different in its function. So it encodes in DNA storage for RNA and protein encoding information and transfers information to next generation of cells, where RNA carries protein encoded information and helps to make proteins specifically. It even can catalyze some reactions. So there are 3 types of RNA. We have messenger RNA, which is when we look at the central dogma, that's where we go DNA to RNA. Transfer RNA is going to be what helps to make RNA to protein. So it's going to be what actually helps carry the information or carry the amino acids to make the protein. And there are RNA that's part of the ribosome specifically. And this is where translation is going to occur. So there are different types of RNA molecules. And shorthand for messenger RNA is mRNA For transfer RNA, it's tRNA, and for ribosomal, it's rRNA. There are other types of RNA, but that is for a more advanced class to discuss those. So I like to think about genes being like a recipe in a cookbook. And when I talked about this earlier in the semester, I compared it to my mom's recipes. My mom was a fantastic cook and baker. I grew up as a child on a farm in Iowa. We didn't have a whole lot of money, so my mom made everything from scratch. So when she passed away from cancer, it was really, really important that I get something that I felt was what reminded me of her, a part of her, and that I really identified with. So I took all her recipe boxes and everything. So all her recipe boxes in her big recipe box has these individual recipe cards. So the whole box itself would be like this cookbook here. Oops. It has every single recipe that she ever made kind of consistently that she didn't have stored in her brain, right? Just like this cookbook does. It has multiple recipes in it. So that would be like our chromosome with a lot of genes on it. But we don't want to cook the entire recipe in the box or all the recipes in the cookbook. We only want to make brownies. So we're going to take out one recipe and then we are going to make a photocopy of that. Now I think about in terms of my mom's box, I don't like really cooking with them because I'm scared I'm going to mess them up. And once I mess them up, they're gone forever. And they make so much, they're very, very close and dear to me because a lot of them are handwritten by her. And so it makes me feel extremely close. So I wouldn't want anything to go wrong with it. So I like to make copies of them. So this is what it's showing here is that copy of that one brownie recipe, right? The great thing is if I make a copy of it and I decide not to make the recipe, I can throw it away. It's no big deal. So if there are signals that say I need to make this protein, but then ends up being so that we don't need to necessarily make it, the cell will just destroy it. And it's no loss, right? Because we have the original recipe or gene still in the nucleus. Now, when we want to make that brownie recipe, we have to basically put things like flour and sugar and salt and cocoa and butter and eggs. All of those individual things would be like the amino acids that we're using here for our protein. So we're going to bring all those different amino acids, make our batter and cook it. And so when we actually get our protein ready or done, that's equivalent to having our brownies ready to be eaten or used however we need to. So the process of making the photocopy, because we stay in the same language, is transcription. And the process of taking that photocopy and making it actually into our brownies is translation. So transcription is a process of taking DNA and creating RNA. And so basically with DNA going to messenger RNA, we are staying in the same type of biomolecule, which is a nucleic acid. So what I want you to do here, and you can see in the description below that there is a video, and you're just going to watch this, and it's going to take you through the central dogma overview. What we're going to talk all the way from DNA to protein, but what we're going to focus on next after you watch this to get an idea what we're about to talk about is transcription. So in transcription, DNA sequences determine the RNA sequence. So when we want to make RNA, what I was talking about earlier, base pairing takes place between the RNA and the DNA molecule. And we use complementary base pairing. So if we look, we do complementary base pairing. Basically, if we have a piece of DNA that has A, C, G, and T, as we know, if we were doing another piece of DNA here, the complementary base pairing would be T and 4C, right? G, N4G, C, N4 thymine A. Those would be the DNA complementary base pairing. Now, when we go to take DNA and we base pair it with RNA, we get U for uracil. C still base pairs with G. That doesn't change from what we saw here. G still base pairs with C, and thymine still base pairs with adenine. So basically, we just replace all our T's in DNA with U's for RNA. And basically, these, if we go through and make base pairing RNA to RNA, we follow the same rules that we did from when we went from DNA to RNA. As you can see, we already have uracil in our RNA there, so it will base pair with adenine. And here again, adenine will base pair with uracil. Now, that's base pairing. How do we make this messenger RNA to begin with? Well, there are 3 steps that take place: initiation, elongation, and termination. So let's talk about each one of these. So in transcription initiation, RNA polymerase gets started. As you remember, enzymes always tell us what we do. So it ends with an ACE. That's how we know what essentially we have an enzyme. And then it's telling us what it makes, which is RNA polymer, right? So if you remember when we were in DNA replication, we had DNA polymerase because it was making a DNA polymer. So what happens here is the gene we want to make. Now, just like if you are someone who's new in the music field, you want your career to get started. You want people to know you and get everything, like maybe you want to get things booked, right? Venues, get on the TV, get on the radio. So you hire a promoter to basically get your career started. So think about this. We have what's known as the very first area here, a promoter region. And this is where our enzyme will bind to start transcription, which makes sense. We're starting transcription, right? Because we're at initiation. So we're starting the process, right? So basically, this is the area on the gene, the promoter region, that RNA polymerase will bind. There are things like transcription factors that help with this binding. So this is initiation in part 1. Now, for initiation, remember that the DNA is always the template to start this, but only one strand will become or be used as the template strand to make messenger RNA. The other strand does not participate at all. The second part of this is elongation. So think about it. We are making a long strand of RNA. So this is where the RNA polymerase is actually making. That's what synthesize means, making the RNA. So the RNA polymerase is going to move along the piece of DNA using complementary base pairing to do this. So if you remember from DNA replication, you had 2 enzymes. You had an enzyme called helicase to unzip. And then you had DNA polymerase to make DNA strand. Here, the RNA polymerase does both. It unzips the strand of DNA, and then it does the base pairing. So he's very efficient. So we do have this base pairing of nucleotides that come in to make this new strain of RNA. The third step we have is termination. And if you get terminated from a job, you're basically getting fired. So you stop working there. That's what happens here. The RNA will reach the end. There is a terminator sequence that tells the RNA polymerase to stop. And once it reaches that end of the gene, it stops, it falls off. Once that termination happens, the messenger RNA is complete. At the terminator sequence, the RNA, the DNA, the RNA polymerase, they all separate from each other. The DNA becomes a double helix again. So the cell has actually produced an RNA copy of that gene. Now, the overall expression of those genes is going to be what it makes at the end. But the way that DNA is packaged regulates gene expression. So in eukaryotic cells, our cells for instance, gene regulation actually starts in the nucleus. Some genes are wound up so tight they can't be transcribed, but other genes are available. And what's cool is once you get that messenger RNA, remember how I said you can't leave your house until you're mature? Well, they're going to go through a process that removes pieces of RNA that doesn't need to be there. And it's going to allow only those pieces of RNA to actually leave. So if you notice, there's two types. There are what's called exons and introns. Introns get removed and stay in the nucleus. Exons exit the nucleus and become what's known as mature messenger RNA. And it's going to move out of those nuclear pores to the cytoplasm. And as I also said about starting transcription, you have what are known as transcription factors, and they help to regulate gene expression too. In eukaryotic cells, many different proteins that are called transcription factors combine to DNA and help with the process of transcription being turned on. This is going to affect the activity of RNA polymerase, altering the rate of transcription. In eukaryotic cells, they can use different combinations of what are called exons. This is what's known as alternate splicing, and this creates different proteins from the same messenger RNA. So you can see here, the introns get cut out and stay in. They really get deteriorated. And then the exons exit, and they become the actual messenger RNA that will be translated to a protein. also have the process of RNA export that regulates gene expression. So certain eukaryotic proteins can actually hold messenger RNA inside the nucleus, making it so that it can never reach a ribosome. So that will also determine whether or not a gene gets expressed or made into a protein. RNA degradation can also regulate gene expression. Degradation means breaking down. So when that messenger RNA exits the nucleus and goes into the cytoplasm, right, that's where it is now, some messenger RNA may be broken down, degraded very quickly. So they can't be translated into very much protein or protein at all. Other messenger RNAs are long-lived and can be translated into a lot of protein over time. So depending on your genes, it can either be translated for a long time or a short amount of time based on RNA degradation. Protein activity can also regulate gene expression. Some proteins are more stable, while others are broken down quickly. Moreover, in order to function, proteins must also be folded properly and reach the correct location. So we were talking about the Creutzfeldt-Jakob and mad cow disease, and as you know, and what we'll talk about later in this talk, is an idea of prions. But first, in order for us to understand what a prion is, we have to know what a protein is and how proteins are made. But as I always say, for instance, I have a shirt on right now. It has a hole at the top so I can put my head in it. It has 2 holes so that my hands and my arms can go through it. And it has a large hole at the bottom so my torso can fit. Now, I could have this shirt made, but if the hole where my head goes through isn't there, it's not going to work as a shirt. So if proteins aren't put together correctly, i.e. folded right, they won't work right. So this is one important thing that we will talk about. So let's review transcription really quick, and you can take some important notes on the things that you feel are the most important in the process. And remember, it is in 3 steps: initiation, elongation, and termination. That's how I remembered them. And this will give you an idea of how this works overall. And this will lead us to the idea of RNA processing. And basically, one more step in order for the RNA to leave the nucleus once transcription is complete, it needs to be processed to become mature. One of those is the introns and exons. So if you think prokaryotic cells, everything happens in the cytoplasm. They don't have a nucleus. So once transcription is complete, they will start translating protein. Now, they can actually have it happen at the same time, as in the process of transcription could be happening, and as it transcribes the DNA at the areas that have already been made to messenger RNA, you can start translation on. It's pretty cool. But in eukaryotic cells, all of this is happening in the nucleus. So there has to be chemical alterations that happen before the messenger RNA can actually leave for translation to start. And one important process is adding a cap and a tail to the messenger RNA. So as you notice, the 5' area that had that phosphate, they're going to add this little structure that's called a cap. And then on the 3', they're going to add what's called a poly A tail. And if you remember, if you've ever heard of anything called polyamorous relationships versus monogamous relationships, monogamous, starting with mono, means 1. Polyamorous means many people in a relationship. So poly A tail just means there's a lot of A's at the end. And this cap and the tail is going to protect the messenger RNA from being broken down once it goes into the cytoplasm. Also, we remove introns, and the exons become the sequence that's going to specify the amino acids. And the introns are sequences in the gene that aren't used for producing a protein. So the introns are removed before it leaves the nucleus. Both of these allow the RNA to be ready for translation or become mature. This processed RNA is now a functional messenger RNA molecule. Once it's completed, that processing, it leaves the nucleus, and then it will bind to a ribosome in the cytoplasm for translation to begin. And this is where we're going to stop for this video so that you can kind of review your notes, look at transcription, and when we start here in a minute, we're going to talk about translation. And translation is, we finish this, right? And now we're starting this, which is the process of taking messenger RNA to proteins. This is happening in the cytoplasm, and the messenger RNA is the messenger from the nucleus on how that's to happen.